Hello, welcome back to another video review. This review is going to be all about endosymbiosis, which is one of my favorite, most interesting theories in biology. It's so special, it has so much support, and so we're going to talk about it today. So before we finish this, I want you to make sure that you are able to define endosymbiotic theory, make sure that it makes a lot of sense to you. You need to be able to discuss the evidence for endosymbiotic theory. That does not just mean list it. You need to understand how it actually directly supports it. So you can't just list, you got to be able to understand and support it that way. And then also be able to describe the advantage of compartmentalization in eukaryotic cells, especially when it comes to transcription and translation. So let's talk about endosymbiosis. So eukaryotic cells probably, almost certainly, arose because of an endosymbiotic event. So we had something like an ancestral prokaryote. So this ancestral prokaryote, it started sort of turning into something that was eukaryotic-like. And so I saw this in my later classes. I didn't see it in my earlier classes that it said eukaryotic here. It really should say eukaryotic-like because we're beginning to see a nucleus. But we have an ancestral prokaryote, this eukaryotic-like cell, and then through an event, an endosymbiotic event, through phagocytosis, an aerobic bacterium was ingested. This aerobic bacterium was ingested and was incorporated into this large cell and ultimately becomes what we now know as the mitochondria. Now, this sounds like it's kind of far-fetched. It sounds like something that shouldn't have happened, or it sounds like it needs a lot of support. The good thing for us is it does have it. So this happened with the mitochondria, and then billions of years later, it almost certainly happened with the cyanobacteria as well to form the chloroplast. So this gave our cells, the eukaryotic cells, a huge evolutionary advantage. Now, you might be asking, okay, about 2 billion years ago, that's when we started to get our nucleus. And that didn't form through endosymbiosis. Instead, that formed from the nucleus folding in upon itself. And it started encapsulating this DNA, which protected the DNA, and that was a big advantage. Then, through endosymbiosis, the mitochondria was incorporated, and then through a second endosymbiosis event, the chloroplast was incorporated. This is pretty cool. Okay, now let's talk about the evidence for this, because it's a pretty extraordinary claim, but it has pretty extraordinary evidence. So let's look. The first thing is that our mitochondria are actually double membraned. So we have lots of membrane-bound organelles, but we don't have very many double membrane-bound organelles. It's really just the mitochondria. And so where did that second membrane come from? Well, recall that it was probably phagocytosis, where this eukaryotic-like cell ingested an aerobic bacteria that resembles now the mitochondria. And basically, when it ingested that, the cell membrane through phagocytosis of the large cell became the second membrane around the prokaryotic or the aerobic bacteria that became the mitochondria. So we can see that in this image here with the cell membrane coming from the large cell, and that would form the outer membrane of the mitochondria. And then the green one here would be the original cell membrane of that aerobic bacteria or the cyanobacteria. And so that forms this double membrane. So the outer comes from the cell membrane of the big cell, and the inner, the green, came from the original cell, the original super, super small cell. So that's pretty extraordinary evidence, because why else would there be two membranes, right? The next piece is that the DNA in mitochondria is actually circular DNA, which is very similar to prokaryotic cells. Now, you'll often see mitochondria, and they're kind of lazy in their drawings, and they'll draw just a linear piece of DNA. Don't fall for that. The mitochondrial DNA is circular DNA, which is very, very similar to prokaryotic cells. Now, first off, why do they have DNA if they weren't originally their own cells? And two, why would it be circular if it wasn't derived from a prokaryote? Because maybe you could argue maybe the DNA came from our nuclear DNA, the stuff stored in our nucleus, but Probably not if it's circular too. So that would be very, very surprising if that was the case. They also have their own ribosomes. So they're able to make their own proteins. Now, nowadays, our mitochondria do not make their own proteins for the most part. There are a few. There are a few proteins that the mitochondrial DNA goes to the mitochondrial ribosomes and actually does translate that protein. But most mitochondrial proteins are actually now coded in our nuclear DNA. And you might be thinking, well, that's pretty crazy. And it is, but you have to remember it's been billions of years. Billions of years. So a lot of the functions of the mitochondria have been sort of taken by the rest of the cell. Okay, the other piece of this is that the ribosomes actually resemble the prokaryotic ribosomes. So the prokaryotic ribosome is 70S. And I told you guys in class, don't worry about this S. This is just a unit. You don't need to worry about it. And you'll also notice that they don't add up. The math here, don't worry about it. So 50 and 30S are the subunit sizes, and they add up, but not really, to 70S. And the eukaryotic is a 40 and 60S subunit that add up, but not really, to 80S. The mitochondrial ribosome is more like this. It's the 70S, and it's very similar to the prokaryotic ribosome. And we also know that when mitochondria divide, they do so in a fashion that is very similar to bacteria in a process called binary fission, and they are approximately the same size as much of prokarya. So prokarya being the domain of bacteria and archaea. So quite a bit of evidence for this really astounding theory in biology. And if you're thinking back to those hallmarks of the cell, which are a cell membrane, ribosomes, DNA and RNA, and cytoplasm, then you are going to realize that the mitochondria actually has all of those on its own, and therefore it could be its own cell. Now I'll tell you now, once again, remember that a lot of the proteins that the mitochondria needs are now actually coded for in our nuclear DNA. So if we took a mitochondria out, it would not be a free living cell on its own anymore. But we have a lot of evidence that billions of years ago, it probably was. And so, taking that all in, we now understand that the mitochondria probably was ingested by a eukaryotic-like cell billions of years ago, and that formed the eukaryote branch. Then, billions of years later, a second endosymbiotic event occurred with chloroplasts to form the plant branch. That means that plants have both mitochondria and chloroplasts. And we have a lot of evidence that they were free living cells because there are a lot of similarities with these mitochondria and chloroplasts. They have ribosomes, they have their own DNA, they're double membraned, they both have their own energy systems, so they both have their own electron transport chains, very important, which indicates that they probably were able to live on their own at one point. Now, the second part of this is talking about the nucleus, which I'll talk a little bit more about in the organelle lecture, but remember that the nucleus is the other defining feature of eukaryotic cells. It is going to be found at the center. It contains our DNA. The nucleolus is going to be the dark region in the nucleus that creates the ribosomes. And this is going to be covered by a double membrane found around the nucleus called the nuclear envelope. Now you might be thinking, well, that is a lot of membranes around my DNA. And it is, so it's a lot of protection, but it also makes it a little bit less accessible. So we need something to access that DNA, and that is going to be our nuclear pores. These nuclear pores are going to be little holes in the nuclear envelope that allows access to the DNA. So these structures are vitally important to regulation. And remember, there's certain processes, transcription namely, that happens in the nucleus. And then the product of transcription is RNA, and RNA is going to have to get out of the nucleus and to a ribosome. And so it's going to have to travel through the pore. Now, during that there's actually quite a bit of regulation that happens. And being able to regulate multiple steps, meaning transcription and translation, is a major advantage of eukaryotic cells to prokaryotic cells. So start thinking about why that might be the case. You might want to do some research on your own, wink, wink, nudge, nudge. But I hope that makes a little bit more sense. Please make sure that you go back to that beginning slide and verify that you can do all of those learning goals before you go and take the exam. Good luck!